Hi, and welcome back to my channel, Learning Biology with Dr. Vanessa. In today's video on the cardiovascular system, we are going to be taking a look at blood vessels, the different types, what they look like, and how they work. Let's first take an overview of the different types of blood vessels. So there are arteries, and arteries are going to be the blood vessels that are taking blood away from the heart. And so I'm going to color code this so that you can understand the difference between blood that's being taken away from the heart, and this would be blood that is oxygenated. So arteries are going to take blood away from the heart versus veins that are going to bring blood towards the heart, and those are going to be deoxygenated. So we show blood that is being taken away from the heart that is oxygenated as red, and blood that is coming back to the heart that is deoxygenated as blue. Now understand that blood is not blue, it is different shades of red, but this helps us to get a visual of what direction blood is being taken in. So we can start again with arteries. And arteries are taking blood away from the heart, and they divide into either elastic or conducting arteries or muscular arteries. As they get smaller, they become arterioles, and then arterioles are going to branch into capillaries. And because this is a closed system, the capillaries is what is going to join the artery side with the vein side. So capillaries are going to then reunite to form venules, and then venules are going to merge into veins, and veins are going to bring that deoxygenated blood back to the heart. So the purpose of the blood vessels is to take that oxygenated blood from the heart, disperse it throughout the body. It gets to that capillary area where the capillaries are going to drop off that oxygen and nutrient to the tissues, and then it's going to converge back into the vein side, and the veins are going to bring back that deoxygenated blood back to the heart. Though there are some differences between blood vessels, there is a basic structure for blood vessels, and that includes 3 layers or tunics. The first one being the innermost layer called the tunica interna. This layer has an active role in vessel-related activities, and this is the tunica or layer that is in direct contact with the blood. The next layer would be the tunica media. This is a layer of smooth muscle that can contract or dilate to help regulate the diameter of the lumen. And the lumen would be the innermost portion that the tunica interna touches, or where the blood would be flowing. And then finally would be the outermost layer, which is called the tunica externa. And this is the layer that is going to help anchor the blood vessel to surrounding tissue. Arteries, as I've already mentioned, carry oxygenated blood away from the heart. They are characterized by a thick tunica media, that layer that contains smooth muscle, allowing these vessels to contract and dilate. They also have a high compliance. This means that the walls of these vessels can easily stretch or expand without tearing. Since arteries, especially those that are closest to the heart, are going to be taking the bulk of the pressure as the heart pumps blood into the arteries, they need to be able to withstand this pressure and have that high compliance so that they're not tearing as the blood goes into the arteries to be dispersed around the body. Elastic arteries or conducting arteries are going to be the largest arteries. The aorta is one of them. These arteries, like I mentioned before, are pressure reservoirs. So they are going to be taking the blood from the heart and allowing it to be dispersed all throughout the body. And so they have to be able to, again, withstand a good bit of pressure and allow some pressure to build up within them so that they can continue to push the blood through the rest of the body. They have a recoil of elastic fibers. So there are elastic fibers in there that allow some recoil. So think of how a rubber band can be stretched and then let go so that it goes back to its original position. This is going to allow these vessels to fill with blood and become that pressure reservoir, allowing for some pressure to build up, and then allowing that pressure that builds up to be able to push the blood again to the other vessels in the body. When that happens, these elastic fibers that are within these vessels allow for the recoil to happen, which is going to allow, one, the blood to be propelled throughout the body, and two, the vessel to return to its original shape. So this is going to include the aorta and the pulmonary trunk. Arteries also include muscular or distributing arteries. These are going to be medium-sized arteries, so the elastic arteries are going to converge into these muscular or distributing arteries. And these medium-sized arteries have a tunica media that has more smooth muscle and less elastic fibers than the elastic arteries that we just talked about before. They are able then to do more with the constriction or dilations that can happen within those vessels, and that is going to adjust blood flow. If they are dilated, more blood can flow through. If they're constricted, not as much blood can flow through. Anastomoses is the union of two or more arteries that are going to supply the same body region. So it's very unique in blood circulation that we just don't have one artery that eventually converges into an arteriole, it eventually converges into these capillaries, but we have anastomoses in several areas where there are going to be many arteries that are coming to the same region, and this is going to allow for an alternate route if necessary. So if there is something wrong with one of the blood vessels, blood can still be supplied to that area because of this union of two or more arteries that are coming into this area. So it provides this alternative route, and we also refer to that as collateral circulation. So again, this area where many arteries can combine so that there is more than one way for the blood to get to that area. So arteries are going to then converge into arterioles. Arterioles are small arteries, about 10 to 300 millimeters in diameter, that are going to then deliver blood to the capillaries. These are also capable of regulating blood flow and blood pressure, where they can have both constriction and dilation. So contraction or constriction of those vessels is going to cause vasoconstriction. When the blood vessel contracts, it causes vasoconstriction, and when they relax, it's going to open up and allow for vasodilation to happen. So this can really help regulate the blood flowing into an area. Capillaries are going to be the smallest of blood vessels with a diameter of about 5 to 10 micrometers. Their primary function is going to be to exchange substances between the blood and the interstitial fluid. So in these areas, the capillaries are going to form a capillary bed, and we're going to get much smaller, basically to where one red blood cell at a time can pass through these areas. And it's going to allow for this exchange to happen where oxygen and nutrients can go into the interstitial fluid, and waste and carbon dioxide can be picked up back by the capillaries to eventually be taken to the heart and removed from the body. Now, capillaries are very thin as well, and they lack both a tunica media and a tunica externa. So they only have a tunica interna, and this allows the substances to pass through just one layer of those endothelial cells and the basement membrane, allowing diffusion, osmosis to occur in these areas that they would otherwise not be able to, because in the arteries that we just talked about, they have a lot of layers. It makes it more difficult. So the capillaries allow for these substances to pass out of them and also for substances to pass back into them. There are three different types of capillaries. The first type is what is called a continuous capillary. Now, in a continuous capillary, we have the endothelial cells, that tunica interna area, and it is going to form a continuous tube. And what you'll also see is that the basement membrane that forms around these cells, this is the lower level of these cells, that basement membrane that's there is also continuous. So we see continuation in both where they are all attached to one another, and you'll see how that differs from the other types of capillaries. We see these types of capillaries in the brain, lungs, skeletal, and smooth muscle, as well as in connective tissues. In a fenestrated capillary, what you'll notice is that within those endothelial cells, that layer that touches the lumen, that tunica interna, we have what are called fenestrations, just like the name implies. And these fenestrations are also pores. They're about 70 to 100 nanometers in diameter. So we see that within the endothelial cells, it has these pores. And as you can imagine, this is going to allow more things to pass through them. However, the basement membrane is still completely intact in these fenestrated capillaries. And again, this is going to just allow a little bit more to pass through in these areas. So we see these types of capillaries in the kidneys, in the villi of the small intestine, where we're going to get nutrients coming into the blood, the choroid plexus of the brain, and the ciliary process of the eye. And we also see them in the endocrine glands because the endocrine glands are responsible for releasing hormones, and they need to be able to find their way into the blood. The third type of capillary that exists is called a sinusoidal capillary. And what you'll see here is that it has much bigger openings within those endothelial cells. So they have these unusually large fenestrations. And not only are they in the epithelial cells themselves, those endothelial cells, but we also see an incomplete or absent basement membrane. So these are going to allow the most things to pass through. As a matter of fact, in these areas, proteins and sometimes even cells are able to come through these areas because of these very, very large openings. So we would see these things in the liver, spleen, anterior pituitary, and parathyroid glands. We would also see them in areas like the red bone marrow when we have newly formed red blood cells that are going to enter the bloodstream. They're going to have to be able to pass through these sinusoidal capillaries. So sinusoidal capillaries are going to allow for much larger things to pass through. So in areas where either of these things need to exit the blood or come back into the blood. And as we get to more specific topics, we're going to talk about how these sinusoidal capillaries play a role. I mentioned the term capillary beds before. And so capillary beds is going to be this area of microcirculation because now we're at the smallest vessels. This is where the interaction between the blood and the interstitial fluid is going to happen. So a capillary bed is a network of about 10 to 100 capillaries, and this is going to connect the arterial flow and the venous return. So again, it's a closed system where the arteries are going to come in with that oxygenated, nutrient-rich blood, bring it to the capillaries. In this capillary area where we're calling true capillaries is where there's going to be the exchange of nutrients and oxygen, as well as the uptake of waste materials, carbon dioxide on the venule side, and then those capillaries converge with the veins on the venule side, and that is what's going to go back to the heart. So since there's this interaction in the capillary bed between the capillaries and the interstitial fluid, we can talk about capillary exchange. Capillary exchange is the movement of substances between blood and the interstitial fluid. And there are going to be three basic methods in how this happens. The first is diffusion, the second is transcytosis, the third is bulk flow. And we're going to go ahead and talk about all three of these. Diffusion is the most important method. In diffusion, substances move down their concentration gradient, or we can also say they move with the concentration gradient from higher concentration to lower concentration. If you remember from past lectures, diffusion is a passive process, which means it requires no energy. In diffusion, oxygen and nutrients are going to move from the blood, those capillary area, to the interstitial fluid, and eventually it will be taken up by the cells of the body. And carbon dioxide and waste are going to move from the body cells to the interstitial fluid back to the capillaries to be taken into the vein side and be taken back to the heart, eventually to be released in different ways. Diffusion can cross the capillary wall through intracellular clefts, fenestrations, or even through endothelial cells themselves. Most plasma membranes cannot cross, except if we're talking about areas where there are sinusoidal capillaries. In those areas, proteins and even blood cells can leave. In the blood-brain barrier, where we have the blood coming into the brain, there are very tight junctions. So in this area, the body limits diffusion, which means even less can come into the brain, and this is a protective measure that the brain has. Transcytosis is the movement through a cell. Now, this happens with substances that are in the blood plasma that become enclosed within pinocytotic vesicles that can enter into the endothelial cell by endocytosis and leave through the other side of the cell by exocytosis. This process is important mainly for large lipid-insoluble molecules that cannot cross capillary walls any other way. An example that would use transcytosis to get through would be insulin and some antibodies from mom to fetus. So this passage is going to be through the cell, being taken into the cell, and then exiting the cell. So coming in by endocytosis, specifically like pinocytosis, and then going through to the other side of the cell by exocytosis. So transcytosis involves going through the cell itself. Now, bulk flow is the third process by which things can move within the capillaries. And bulk flow really describes this whole process as to how things come out of the capillaries and things move back into the capillaries. And bulk flow also relies on diffusion. And this can be really complicated, and there are pressures involved within this process as well. I'm going to give you a little overview of what bulk flow is in this video, and then I have another video where I go into this in more detail so that you can understand it better. So for all intents and purposes for this video, I want you to understand that bulk flow is a passive process in which large numbers of ions, molecules, or particles in a fluid move together in the same direction. And what we're talking about here is we're talking about things going from the capillary bed on the artery side into the interstitial fluid, and then things going from the interstitial fluid back into the capillary bed, but on the venule side. And the movement is going to occur from an area of higher pressure to an area of lower pressure. Bulk flow is important for regulation of relative volumes of blood and interstitial fluid. We don't want to push everything out of the capillary, but we do want things to go into the capillary. And at the same time that things are going in to the capillary, moving out of the capillary, things are moving back into the capillary as well. So we have two big processes that are happening during bulk flow. We have filtration. And filtration is the pressure-driven movement from the capillaries into the interstitial fluid. So filtration is going to happen on the arterial side, where we're going to move things from the capillaries into the interstitial fluid, and then we're going to have reabsorption, where there's going to be a pressure-driven movement from the interstitial fluid back into the capillaries. There are two pressures that promote filtration, And then there are going to be two pressures that promote reabsorption. Again, I'm just going to mention these, tell you where they come from, and then you're going to watch another video later where you're going to learn more about bulk flow and how bulk flow works. But I want you to have a base knowledge and start to build on that, because this can be confusing. So two pressures that promote filtration. Now remember, filtration again is going from the capillaries on that arterial side into the interstitial fluid. So we have blood hydrostatic pressure, abbreviated BHP, and this pressure is generated by the pumping action of the heart. You also know it as blood pressure. This pressure pushes fluid out of the capillaries into the interstitial fluid. The other pressure that is going to help promote filtration is interstitial fluid osmotic pressure, abbreviated IFOP, and this pressure pulls fluid out of the capillaries into the interstitial fluid. So blood hydrostatic pressure is a pressure that we are going to find within the capillary itself, and the interstitial fluid osmotic pressure is going to be a pressure that we find in the interstitial fluid. And then, of course, there are those two pressures that promote reabsorption. And reabsorption is, well, we got this fluid out into the interstitial fluid, into the interstitial area. We can't just leave it all there. We have to bring it back into the capillaries. And so the pressures that promote reabsorption are going to promote that fluid going back into the capillaries on the venule side. The first pressure is blood colloid osmotic pressure, abbreviated BCOP, and this is a pressure that is found within the capillary itself. This pressure is due to the large proteins that are found in the blood plasma that cannot leave the capillaries. And so this pressure is going to actually pull fluid from the interstitial spaces back into the capillaries on that venule side. And then there's the interstitial fluid hydrostatic pressure, abbreviated IFHP. This pressure is found in the interstitial fluid itself, and it is going to push the fluid from the interstitial spaces back into the capillaries, again on the venule side. Once the interaction within the capillary bed has happened, we have that fluid that's moving into the venule side now. So those capillaries in the capillary bed are going to unite and form those venules that I've been referring to, and those venules are going to converge and form veins. Now, veins are unique in that they have no elastic layers. They have a very large lumen. The lumen is going to be larger than the lumen that we see in arteries. Arteries have a lot of pressure. They have to be able to push that blood throughout the body. The veins, on the other hand, are going to just be moving that blood back to the heart, so they have a larger lumen. They also have valves. We do not see valves in the arteries, but we do see valves in the veins because this is going to help aid in venous return. Remember, veins have to come in large part against gravity. Think about blood coming from your feet back to your heart. So in large part, it has to move against gravity. And because of that, the veins have these valves that can open and close and help prevent backflow. So instead of blood going backwards, the valves can close and allow the blood to reach the heart. Venous return is then the act of blood flowing back to the heart through these veins within the circulatory system. And this is going to be done by the pressure generated by contraction of the left ventricle of the heart. Now, the left ventricle of the heart generates a lot of pressure because it has to overcome the pressure in the aorta to allow blood to move all over the body. And it's going to generate this pressure, and this pressure is going to allow blood to flow back to the heart. Besides the heart, there are two other mechanisms that must pump blood from the lower body to the heart. This includes the skeletal muscle pump and the respiratory pump. Let's talk about these a little bit more so that you can understand how they work. The skeletal muscle pump, just like its name implies, works with the contracting action of the muscles. So when you're standing at rest, if we're looking at the leg muscles, the back of the leg, the calf muscles, when you're standing at rest and those muscles are relaxed, both of the venous valves are going to be open, and this is going to allow the blood to flow towards the heart. Now, there are going to be two valves, the proximal valve, which is going to be closer to the heart, and the distal valve that is going to be further away from the heart and closer to the foot. That would be down here that I didn't draw. So standing at rest, these valves would both be open, allowing blood to make its way up towards the heart. Contraction of the leg muscles then compresses the vein. This pushes blood through the proximal valve, and this action is known as milking. So the muscles contract, this compresses the vein, and this is going to push the blood up through the proximal valve that is closer to the heart, and the distal valve is going to close. This prevents backflow. When the muscle relaxes after contraction, pressure is going to fall. This is going to cause the proximal valve to close. The distal valve opens because blood in the foot is going to be higher than in the leg, and the vein is going to fill with that blood from the foot. And then eventually, as pressure differences even out, we are going to go back to complete rest, where both the venous valves are going to be open, allowing the blood to continue to flow towards the heart. The respiratory pump aids blood flow through the veins of the thorax and the abdomen. During inhalation, the volume of the thorax increases as the diaphragm moves downward, which causes a decrease in pressure in the thoracic cavity and an increase in pressure in the abdominal cavity. Then the abdominal veins are compressed, and a greater volume of blood moves to the thoracic veins and then into the right atrium of the heart. When pressures reverse during exhalation, valves in veins prevent backflow of blood from thoracic veins to abdominal veins. The largest portion of blood at rest, as you can imagine, is in systemic veins and venules, with about 64%. These are referred to as blood reservoirs. Venoconstriction, or constriction of the veins, reduces the volume of blood in these reservoirs and allows greater blood volume to flow where needed. We can go ahead and conclude this video by looking at the difference between arteries and veins. Now, arteries are going to conduct blood away from the heart. That blood is going to be oxygen rich, while veins are going to conduct blood towards the heart, with that blood being oxygen depleted, which we refer to as deoxygenated. The general appearance of arteries is that they are rounded, whereas veins are irregular, often collapsed. And this in large part has to do with the wall thickness. Arteries have a thick wall, and veins have a thin wall. And the thicker wall of the arteries allows for it to have a better structure, where veins are more flimsy because they're thinner. And so that's going to give rise to their different appearances. The pressure in arteries is high. Again, arteries are bringing blood away from the heart throughout the entire body. So there's going to be a lot of pressure in arteries, whereas in veins, there's going to be low pressure. And as I already mentioned, there's going to be higher oxygen concentration in arteries and lower oxygen concentration in the veins. And the difference, though, is that in the heart blood flow itself, when we talk about the pulmonary trunk is actually an artery, and that is going to be the exception to the rule. The pulmonary trunk has lower oxygen because that is the artery that brings blood away from the heart, but it's bringing blood to the lungs to be oxygenated. And then the other reverse that we have is that those veins that go from the lungs back to the heart, those veins are going to have high oxygen because they're coming from the lungs back to the heart. And then eventually that blood is going to be pumped out through the left side of the heart. So there are a couple exceptions to the rule. And then finally, we do not see valves in arteries, but we do see valves in veins. And again, this is because in veins, we are going to be bringing that blood from the body back to the heart, and a lot of that is going to have to go against gravity, and those valves are going to help to prevent the backflow of blood and help it to reach its final destination, which is going to be the heart. Thank you so much for watching my video. If you liked my video and its content, please make sure to support my channel by subscribing and liking this video. Teachers, I have notes that go along with this video. If you'd like to access them, you can click on the link below in the description box that links to my teachers page that has not only notes for this video, but also other great resources for anatomy and physiology.